In this video, we're going to talk about homologous recombination. We'll do a quick review of homologous recombination. Homologous recombination is a strand exchange between a pair of DNA that are similar or identical. The match need not be perfect, but it must be very close. It takes place only between DNA duplexes that have extensive regions of sequence homology, and it is conserved in nearly all organisms. And it generates novel combinations of genes during meiosis. It can accurately repair double-strand breaks, whereas non-homologous end joining leaves mutation, so it is a major way to repair double-strand breaks. It occurs just after DNA replication, when two daughter DNAs lie close together. Let's look at the process. Firstly, the 5' ends of the damaged DNA are chewed back to produce a 3' overhang. Secondly, strand invasion occurs. The 3' end searches for its homologous sequences carried out by the RAG-A or RAD51 protein. And extension of invading strand. So the extension occurs by using the undamaged strand as a template, and after that, the strand is released. The complete synthesis of damaged strands by utilizing the repaired strand as a template. And finally, the DNA ligation. If you look at the figure, the figure is so perfect, I don't need to further elaborate. The detailed procedure mediated by RAG-A or RAD51. It is called RAG-A in E. coli and called RAD51 in all eukaryotes. Okay, so firstly, the nuclease MRE11 degrades the 5' ends of the damaged DNA. And secondly, the ATP-bound RAG-A or RAD51 binds cooperatively to the invading single strand, forming a protein DNA filament that forces the DNA into an unusual configuration. Thirdly, groups of 3 consecutive nucleotides are held as though they were in a conventional DNA double helix, but between adjacent triplets, the DNA backbone is untwisted and stretched out. Protein DNA filament then binds to duplex DNA in a way that stretches the duplex, destabilizing it and making it easy to pull the strands apart. And finally, the single strand probes the undamaged strand, and probing occurs in triplet nucleotide blocks. If a triplet match is found, the adjacent triplet is probed, and so on. RAG-A or RAD51 hydrolyzes ATP to be disassembled from the DNA, but note that probing does not require ATP hydrolysis. Let's look at the figure. So you can see that the DNA is stretched out in RAG-A bound form. The use of accessory proteins is also important to note. RAD52. RAD52 is a donut-shaped protein which has a deep groove that enables the single-stranded DNA to run along the protein surface. It helps to load Rad51 onto single-strand DNA to form the nucleoprotein filament, and it acts later to reform the double helix and complete the homologous recombination reaction. This is the structure of a portion of the Rad52. You can see the deep groove and a DNA running through it. The use of accessory proteins is also important to note. BRCA1, which is an important protein, regulates an early step in broken end processing. Without it, the ends are repaired by the non-homologous end joining pathway. BRCA2. BRCA2 binds to RAG-A or Rad51, preventing its polymerization on DNA, thereby maintaining it in an inactive form until it is needed. Upon DNA damage, BRCA2 helps to bring RAG-A or Rad51 to the sites of damage and, once in place, to release it in its active form onto single-strand DNA. Holiday junction. Holiday junction is a reaction intermediate of homologous recombination. The heteroduplex region extends because of branch migration. It might form a double holiday junction. Branch migration is the moving of the branch point by the continuous breakage and rejoining of DNA, and it is catalyzed by the enzyme ROV-A and ROV-B in E. coli. ROV-B resembles helicases, hydrolyzes ATP to spool DNA rapidly through the holiday junction, extending the heteroduplex region. ROV-A coordinates this movement, threading the DNA strands to avoid tangling. Let's look at the figure. You can see the heteroduplex region is extended because of branch migration. You can see the chicken foot shaped holiday junction on the center and the two proteins ROV-A and ROV-B. Repairing replication fork with a single-strand nick. The figure that I'm going to show you next is so perfect. I don't need to elaborate more on top of that. As you can see, there is a single-strand nick just ahead of the replication fork. The replication fork eventually breaks, and nuclease degrades the 5' end of broken strand, and then strand exchange occurs, and then strand extension, and then you can follow the step drawn here. Regulation of homologous recombination. It is really important to regulate the homologous recombination reaction, but sometimes cell errantly uses homologous chromosomes instead of sister chromatids, which can lead to the loss of heterozygosity, which means the convergence of maternal DNA to the paternal or vice versa. The processing of the broken ends is coordinated with the cell cycle. The nuclease enzymes that carry out this process are activated by phosphorylation only in the S and G2 phases of the cell cycle when there are daughter duplex that can serve as a template. Enzymes that catalyze recombinational repair are dispersed throughout the nucleus in an inactive form. In response to DNA damage, they rapidly converge on the sites of DNA damage, become activated, and form repair factories, where many lesions are brought together and repaired. The homologous recombination during meiosis. The homologous recombination produces both crossovers and non-crossovers during meiosis. Most of the recombination event, which resembles the reaction for the double-strand break repair, produces non-crossovers, where only the region near the site of the double-strand break is altered. A crossover is formed by cleaving a double Holiday junction, thereby swapping the upstream and downstream of the chromosome. The process. Let's just look at the figure. But before then, I'll just add a little description. Firstly, spole 11 in budding yeast breaks both strands of the DNA. Like a topoisomerase, spole 11 remains covalently bound to the broken DNA. MRE11 degrades the 5' ends bound by spole 11, removing it along with the DNA and leaving protruding 3' single-strand ends. Okay. The step here is quite the same as what we have discussed already, but yeah, and here. But what we have to note is this part. And this part forms a double Holiday junction, and it creates a crossover. So the next thing that we're going to discuss is the crossover control. Crossovers are evenly distributed along the chromosome. Crossover in one position inhibits crossing over in neighboring regions. Every chromosome undergoes at least one crossover. Roughly 2 crossovers per chromosome occur during each meiosis, one on each arm. As you can see, this is a non-crossover, and this is a crossover, right? And this is the site where gene conversion occurred, and this is the site where crossover occurred. Gene conversion. Gene conversion is the conversion of a segment of a DNA from maternal to paternal and vice versa by mismatch repair system. Recombinant DNA contains heteroduplex regions where maternal and paternal DNA are swapped. So in the heteroduplex region, if the contents of their sequence are different, mismatched base pairs will be included, which may lead to DNA mismatch repair. Consequently, not both parents are making the same genetic contribution to an offspring. Okay, so the heteroduplex generated during meiosis covers site in gene X, where red and blue alleles differ. So the mismatch repair system cuts the portion of blue strand, and the DNA synthesis fills the gap, creating an extra copy of the red allele of gene X. So for now, we're finished, and thanks for watching. Bye.